Hi everyone, welcome to my channel. Welcome to the second video of structure and function of eukaryotic cell. We are discussing here about the structure and function of different cell organelle of eukaryotic cell. Now we are going to start with the main organelle of eukaryotic cell, that is nucleus. So let's start. Nucleus. It is a double membrane bound dense body which controls all cellular metabolism and encloses the genetic information of the cell. It is also called the brain of cell or head office of the cell. Nucleus as a cell organelle was first described by Robert Brown as early as the year of 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. Robert Brown, 1831, studied orchid root cells and first of all described nucleus. Hammerling studied Acetabularia and proved the importance of nucleus in its heredity and growth. A true nucleus is absent in bacteria, blue-green algae, a few dinoflagellates, and mature RBCs among eukaryotes. These instead possesses an incipient or false nuclei. The cells may be uninucleated, binucleated, or multinucleated depending upon the number of nuclei present in them. The shape of a nucleus may be disc-shaped, spherical, ovoid, bilobed, multilobed, kidney-shaped, C-shaped, or a string of beads, etc. Position of the nucleus varies from cell to cell, but it is mostly found in metabolically active regions. For example, nucleus occupies central position in embryonic and glandular cells, while peripheral in fat cells. In the structure of nucleus, the main four parts are found. Number one, the nuclear membrane, and then the nucleoplasm, and main part of the nucleus is nucleolus and chromatin network. The nuclear membrane, it is also called as a nuclear envelope or karyotheca. It is composed of two membranes, that is the outer and the inner membrane. These are separated by means of a perinuclear space. The outer membrane of nucleus is continuous with endoplasmic reticulum and it may bear ribosomes also. Nuclear membrane contains octagonal nuclear pores. These pores are formed by the fusion of outer and inner membranes. Callan and Tamlin in the year of 1950 were the first to observe nuclear pore in the nuclear membrane. These pores control the inward and outward movement of different materials such as mRNA, nucleoproteins, etc. from the nucleolus. The nuclear pores are enclosed by circular structures called as annuli. The pore and annuli together constitute the nuclear pore complex or pore baskets. And the second is nucleoplasm. Nuclear sap or nucleoplasm is the fluid part of a nucleus. It majorly contains DNA and associated proteins along with water, ions, enzymes, soluble molecules and nucleotides. It is considered as the main site for the synthesis of DNA. The DNA and associated proteins are visible either as chromatin or as chromosomes. And the main part of the nucleus is nucleolus. The nucleolus was first recognized by Fontana in the year of 1874. It is a spherical membraneless body within the nucleus. Its constituents are RNA, DNA and proteins. The nuclei are formed from specific sites on chromosomes. These sites are called nuclear organization regions. A nucleolus can be best visualized at interphase. It is mostly found in cells that are involved in protein synthesis. It is absent in yeast, bacteria, some alga, embryonic cells, sperm cells, and erythrocytes. Nucleus is the most important site of ribosomal RNA synthesis, which comprises about 80% of total RNA content of the cell. Thus, it is the site of ribosome synthesis. The fourth part of the nucleus is chromatin network. The chromatin, meaning colored fibers, are fine fibers composed of DNA, some proteins, and transcribing RNA. The proteins associated with chromatin are mainly histone proteins. These are positively charged basic proteins mainly composed of lysine and arginine. However, in certain types of sperms, stones are replaced with protamines. During cell division, the chromatin condenses and becomes thick enough to form specialized structures called chromosomes. It was observed that when chromosomes are stained with basic dyes like acetocaramine or fuelgen stain, two types of regions can be observed: heterochromatin region and euchromatin region. Now, the function of nucleus. Nucleus stores genetic information in the form of genes. Second, nucleus tends to act as the main controller of all the cell activities. DNA replication, DNA transcription, and protein synthesis take place in the nucleus. And third, it possesses all the information required for growth, metabolism, reproduction, and development of an organisms. And the fourth one, nucleus provides a site for the synthesis of ribosome and RNAs, and it helps in the process of gene expression and cell differentiation also. Next cell organelle is mitochondria. Mitochondria are called powerhouse of the cell. These organelles are involved in cellular respiration, that is oxidation of cellular nutrients and energy generation of the cell. Richard Altman in the year of 1890 discovered mitochondria. Mitochondria name proposed by C. Benda in the year of 1897. These are unique organelles as they are formed by binary fission and contain their own DNA. Mitochondria are mostly found in actively metabolizing cells and tissues, example like wings of insect. In the chemical composition of mitochondria consist of 70% protein, dry weight, and 25 to 30% lipids. Among lipids, 90% are phospholipids, the rest 10% includes carotenoids, cholesterol, vitamin E, etc. The shape of mitochondria is highly variable. It ranges from short rod shape to elongated filamentous form. These organelles have the ability to change their shapes according to the need of cell. This ability to change shapes is known as pleomorphism. In the structure of mitochondria, these organelles are bounded by two membranes, namely the outer and the inner membrane. These membranes are separated by space called intermembrane space, which is 40 to 70 angstrom thick. This space is filled with matrix. The outer membrane of mitochondria is smooth and freely permeable. It consists of phospholipids and cholesterol. It also contains porine protein, which acts as transport channels. This inner membrane is folded into numerous enfolding like crista. The presence of crista increases the surface area of the mitochondria. It is semi-permeable and rich in enzymes and carrier proteins. Several knob-like structures are also visible over crista. These structures are called oxysomes or F0 to F1 particles or ATP synthesis, complex or subunits of Fernandez-Moran or elementary particles. They are found to be involved in the release of ATP. The space inside the inner membrane is also filled with matrix. This matrix contains dense granules along with ribosomes and mitochondrial DNA. Out of the total enzymes present in a cell, mitochondria also contribute to 70%. The enzymes of Krebs cycle are present in the matrix of mitochondria. Enzymes required for ETS and oxidative phosphorylation are found in crystal. Mitochondria is called a semi-autonomous organelles as it has its own protein synthesizing machinery. The function uh, formation of ATP through oxidative phosphorylation. In the process, ATP is produced through Krebs cycle and electron transport system. Number 2. Intermediate proteins of cell respiration are used in the formation of steroids, cytochromes, chlorophyll, and amino acids such as glutamic acid, aspartic acid, nucleic acid, etc. Mitochondria also regulate the calcium ion concentration inside the cell. It provides a site for thermogenesis, that is the production of heat in body. Number 4. Eunucleus with a mitochondrial cloud and Golgi bodies control vitellogenesis in eggs. Number 5. During spermiogenesis, required energy is supplied by mitochondria. Thanks for watching.